Okay, we are on page 77 of our handbook, looking at how can DNA be used by the cell to make a protein. So A, the importance of protein synthesis. So every inherited trait, meaning all of the things about you that you see, is controlled by one or more proteins. Protein synthesis is the process that makes those proteins. Remember back from Module 1, synthesis means to make. So this is just how we're making proteins. 2, each cell must produce different proteins, and that's based on the function or the job of that cell. So for example, only blood cells would need to produce hemoglobin. Remember, hemoglobin is what carries oxygen throughout the body. So again, when we're talking about a trait is really just what you see. And a protein is what controls your traits. So your proteins control traits. So all of biology is based around what we call the central dogma, meaning this is what everything in biology and life really rotates around. So the first thing here is that the central dogma is that your DNA controls your production, that synthesis of proteins. So there's a few terms to remember when we're talking about this. A section of DNA, which is your blueprint for a protein, is called a gene. So genes are just small pieces of DNA. Every gene has a specific sequence or order of nucleotides. We represent that by writing the order of nitrogen bases. So for example, ACG, CCA, TGC, TAC. So again, the order of the bases is what determines your trait. And every 3 bases, so every 3 letters, is called a codon. A codon is just like one word in a sentence. And when you put together those words or codons, you would get your protein, which is the equivalent of a sentence. So those 3 nucleotides, those 3 letters, are making your codon. And one codon controls the placement amino acid. Remember, amino acids are what proteins are made of. So you put all of those amino acids together, and that will make you your protein. So again, your proteins are made of those amino acids. Every codon, every 3 letters, will direct your cell to make a specific amino acid where the protein is built. So for example, if you have the codon CAA in DNA, it eventually codes for your amino acid called valine. If this codon was the 3rd codon in a gene, that means valine would be the 3rd amino acid. And your central dogma is basically you start with DNA, it gets turned into RNA, which is called transcription, and that RNA gets turned into a protein, which is called translation. So your central dogma is literally DNA gets transcribed into RNA, and RNA gets translated into a protein. So we're about to go into the specifics of how this actually happens. So the first step in protein synthesis is called transcription. The whole purpose of transcription is that we are rewriting the DNA code as messenger RNA. We call it mRNA for short. And this has to happen because your DNA is way too big to leave the nucleus, so it cannot diffuse through the pores of the nucleus. So RNA is its messenger. It sends its instructions. So think about if Mr. Legrand is our nucleus, he doesn't always go to every single teacher to give instructions. He might send out an assistant principal to give us instructions instead of him leaving his office. Same idea here. That mRNA copies the DNA when your DNA unzips one section called a gene. So just like with DNA replication, you have an enzyme that's going to unzip your DNA. One gene makes one protein. So that means every single trait has its own gene. You've got millions of genes in your body. Your messenger RNA is made one nucleotide, one letter at a time, and the DNA is what we call a template. All RNA has a different sugar, ribose, instead of deoxyribose, which cannot bind to thymine. So RNA uses a different base, which we call uracil. So uracil is what is different. Sorry, I meant to highlight that. So uracil is used instead of thymine. So if the DNA was CTA, your mRNA would be GAU. So this just means your base pairing is different, and I'm just going to write that out over here for you. Most of your rules are similar, except the only difference now is anytime you see an A, it would become a U if you're turning it into RNA. The T would still become A, the C's would still become G's, and the G's would still become C's. So the only difference when you're transcribing is now when you pair, A's would become a U instead of a T. Once this mRNA is made, it leaves the nucleus through a small opening called a pore, just like your skin has pores. So again, this is all happening inside the nucleus. DNA is turning into RNA, and that RNA leaves the nucleus, and your DNA will get rezipped by more enzymes. So here we're going to highlight the mRNA. You know you have mRNA when you see the letter U. So if you look at this strand here, and it's already labeled as mRNA, this is what we're highlighting. And you know this is RNA because I see the letter U right there. If I see T, that's how I know it's DNA. So this diagram, all that's happening here is your DNA is being unzipped and transcribed into M, messenger RNA. Once that RNA leaves the nucleus and goes into the cytoplasm, that's where translation happens. This is when you use the mRNA to build a protein. So in your cytoplasm, this is happening specifically on the ribosome. If you remember back to Module 2, ribosomes are the site of protein synthesis, so this is where your proteins are actually assembled. Your ribosomes are made of RNA, which is specifically rRNA or ribosomal RNA, and proteins. So this step, translation, the whole purpose is to build a protein, and we're going to talk about how that happens. So the start codon, just like in a race you have a start sign, the start codon is always AUG, and it attaches itself to a ribosome. That ribosome attaches to the mRNA, and it helps put the amino acids together. We have transfer RNA, and transfer RNA, I'm going to circle it over on this diagram. This is transfer RNA. Sorry, bear with me. tRNA. This literally carries the amino acid over, and it binds with the mRNA. So this guy comes along, attaches to the ribosome, deposits an amino acid. The amino acids, you see these little circles? Those are your amino acids. They keep building and building until you get to a stop codon. These amino acids are bonded in order, and they're attached by what we call peptide bonds. The whole protein is called a polypeptide for many peptide bonds. And again, when you get to a stop codon, your protein is made and it's done. So again, with translation, you're just making the protein. So the tRNA brings the amino acid over, deposits it. The order is what determines the protein. So the protein is all of these little circles, the actual assembled amino acids. In this diagram, what's happening is translation. So the protein is being assembled. Okay. So the result of protein synthesis, literally, it's called protein synthesis because you have a protein at the end. So your protein is made. That is your end result. So all of your cells respond to their environment by making different types and amounts of proteins. So we can have proteins that are structural, meaning they could be part of cell materials. Remember, the cell membrane has proteins in it to help with transport. Or they can be functional. They have a job. So remember, your enzymes, they help speed up reactions. You have hormones, which send chemical messages, or just the chemicals that are used in cellular chemistry. All of your cells have the same DNA, but how they're expressed, how we see them, can be different. So you, as a multicellular organism, were an undifferentiated mass of cells, meaning your cells didn't have a job yet. When your cells start expressing different genes, we call that differentiation. Think it has the word different in it. This is how your cells become the different things. So only specific parts of the DNA are activated. The reason for that is it would waste a lot of energy if all of your DNA was turned on at the same time. So in a nerve cell, only the genes that are related to your brain cells would be turned on. Everything else would be turned off. We call that turning on or turning off expression. Once a cell becomes a specific type, it can't be changed. So muscle cells will stay muscle cells, nerve cells will stay nerve cells, and so on. Gene regulation is how we figure out what genes will be expressed. This is affected by your environment. So you may have too many proteins, too few, or not the right amount. So for example, cancer is when your gene regulation goes out of control, too much mitosis. Basically, your genes control themselves. That's a Stern-Gerr process. And D, every individual in a sexually reproducing population has slightly different sequence of nucleotides, meaning a different order of the A's, T's, C's, and G's. And that's what leads to the different proteins. That produces different traits, which we call variation. So for example, we have different eye colors, skin colors, heights. All of that is because our base sequence, that order of letters, is different. So gene regulation can lead to cell specialization because we have regulation determining what genes are expressed. Which again just means your genes control what is going to get turned on and off in specific cells. Okay. Thank you for listening to my voice for this long. I hope this cleared things up a little bit. If you have any questions, please make sure that you are checking in with me right now. You should complete the check yourself and then get a stamp.